So thank you so much. My name is Lynne ZYDOWSKY. I'm the Chief of Science for Alexandria. Thank you for being here today. Beautiful day in this beautiful space. The title of our session is Immunology and Inflammation, or better known as I&I, a cornerstone of treating human disease. So a big thank you to the Galien. Thank you, Bruno. Thank you, our co-chairs, Mike Rosenblatt and Phil Sharp. We're truly honored to be participating again this year, and a special thank you to Roy Vagelos for his everlasting vision. I'm sorry he isn't here with us today. So, to get started, the field of immunology has emerged as foundational in how we approach a wide array of human diseases, from cancer to autoimmune disorders to neurology, including Alzheimer's, Parkinson's, and even neuropsych. Recent breakthroughs in our understanding of the immune system are leading to revolutionary therapies and opening new frontiers in medical science. And the latest developments, from engineered cell therapies to novel and modulatory drugs, the promise to transform the treatment landscape for millions of patients worldwide. So I would like to give brief bios and introduce our esteemed guests. I'm going to start with you, Steve. Steve HARR is the President and Chief Executive Officer of Sana Biotechnology. Prior to co-founding Sana, he served as the CFO and the head of corporate development for Juno Therapeutics. Juno was a collaboration between the Fred Hutch and Memorial Sloan Kettering. It was one of the first companies to commercialize cell-based therapies, and he was CFO until its acquisition by Celgene in 2018. Prior to joining Juno, he was at Morgan Stanley. He was managing director and head of biotechnology investment banking, and previously a biotech research analyst and co-head of the global healthcare research. I have next to me Dr. Tom Lynch, and I should say Dr. Steve HARR. Everyone with me today is a doctor, a medical doctor. Dr. Tom Lynch, who is the president and director of the Raisbeck Endowed Chair at the Fred Hutchinson Cancer Center. Tom is a world-renowned scientist, he's a highly respected oncologist, and he's a successful leader of the National Cancer Institute designated comprehensive cancer centers. Before taking over as the Hutch's sixth president in February of 2020, Dr. Lynch was the chief science officer at Bristol Myers Squibb, BMS, and prior to that, he held leadership roles as CEO of Massachusetts General Physicians Organization, and he was the director of Yale Cancer Center. Early in his career, he was part of the first research team to discover how targeted therapies could dramatically change outcomes for lung cancer patients with mutations in the EGFR gene. And seated in the middle is Dr. Priya SINGHAL. I hope I'm saying your last name right, who is the Executive Vice President and Head of Development at Biogen. She's an accomplished physician, scientist, business C-suite leader, committed to developing drugs across diverse therapeutic areas that transform the lives of patients and their caregivers. In this role, she has accountability for the entire Biogen clinical and marketed portfolio. Prior to that, she was head of R&D at Zafgen, and prior to that, she had roles in increasing seniority at Vertex and served as head of their Hep C portfolio, as well as HCV and the cystic fibrosis portfolio. So thank you all three. Okay, let's get started. Over the last decade, we've seen increasingly challenging implications emerge for the human immune system across a growing list of disease areas, as biopharma has coalesced around immunology as a central pillar of disease. Between 24 to 50 million Americans have an autoimmune disease, and 4 out of 5 diagnosed are women. Immunology was mission critical in 2020 against the backdrop of the COVID-19 pandemic, as well as amidst a cresting wave of interest and investment into immuno-oncology. 4 years later, we find ourselves in the middle of another gold rush in immunology and inflammation, characterized by breakthrough scientific discoveries, unprecedented clinical success for previously intractable intractable immune disorders, and rapid growth in venture and R&D investment into new modalities that target the immune system to fight disease. I saw this quote, I thought it was so great. It said, "Move over oncology. I will write the next big area of innovation." So I have a question for all of you, but I'm going to start with you, Priya. The field of immunology has been around for a very long time, but why now does it feel so new?
Thank you, Lynn, and wonderful to be here with this esteemed panel for this very important conversation. Good day to everyone. I think that my belief is that when you are looking to solve the highest unmet needs for patients, there's got to be a tipping point. And I've seen this across different therapeutic areas as an observer, as a participant. And today, I think we are at that tipping point for immunology. And I'll just give you a few examples that have made me a believer. The first is really when you go back to 1908 and you look at the Nobel for Metchnikoff and Ehrlich. And then you fast forward to 2017, another Nobel in the same area. So it's kind of a lot of research that's gone into the space for immunological mechanisms, tools, and more. So why now? What have we seen in the last decade that has just changed how we think about it? And I'll just start with a few examples. I think till 2011, we had never seen a phase 3 for metastatic melanoma succeed. And that gave us Yervoy, a checkpoint inhibitor, CTLA-4, and that changed everything. Fast forward to 2014, we saw the PD-1s again for checkpoint inhibitors, and they kind of changed the landscape for cancer, myeloma. And then in 2017, just 2 years later, 3 years later, we saw the CAR-Ts for hematological malignancies. And I think that we're going to continue to see this because of three things that have come together. We understand more about the immune mechanisms. We know that this is a mobile system and cells go from place to place in the body. We can also measure them, and our ability to measure them has gotten far more superior and sophisticated. And three, I think today we know that it's possible, at least this is my belief, that cancer could be at one end of the spectrum of sort of the immune dysregulation, and autoimmunity could be at the other end. So in cancer, we're trying to unlock personal cells to come back and sort of address the cancer. And in autoimmunity, we're trying to suppress them. So I think that the lessons we've learned from 2010 to today with immuno-oncology and immunotherapy in oncology can be really well applied. I also think this is a very high unmet need for patients. 3 times as many patients have autoimmune diseases, and the funding and investment in this area is 7 times lower. So I think and I hope that this is that decade.
I don't know. Steve? Thank you.
Sure. Maybe first thought it just struck me when there's a title of a panel and it's on I&I or inflammation and immunology, and none of us are immunologists, as they talked about it. I think that
which could be a problem, Steve.
It's either a problem or an indication of really the breadth of areas that the immune system touches on and the pathology that it introduces. Because none of us are experts, but clearly we've had to learn enough to be interlopers because it's such a big part of every single disease. I think that when I think about why now, I'd be very careful because it isn't as if we haven't been making progress, right? And for those of us who trained in medicine, and we're not that old, maybe I am getting older, but in the 80s and 90s, we just had one way to treat these immunologic diseases, and it was with sledgehammers, right? You would go and really wipe out and obliterate the immune system. And very frequently, the patients were hospitalized as much for the toxicity of the therapy as they were for their actual underlying disease. And beginning in the 1990s, we actually began to have targeted therapies, right, where we could go after single pathways or single molecules. And that led to a flurry of innovation over 20 or 30 years where there were basically mostly antibodies, but some small molecule-based therapies going after single targets. And we were able to chronically hold these diseases at bay. And these are some of the largest drugs in the history of our industry, right, or the pharmaceutical industry. But then I think something really profound happened 3 years ago, and that was data from a gentleman in Germany named George Schett. And George Schett treated the first patient with lupus with a CAR T cell. And for the first time, very similar to what Priya outlined in cancer, you went from big bombs to targeted therapies to curative intent, right? And that is just a transformation for patients, this idea that you could live forever and that disease might be gone and not something you wake up every day thinking about. And I think we're doing the same thing in immunology today. We're moving from big bombs to targeted therapy to now this idea of curative intent. And if we can, particularly, these immunologic disorders hit a lot of young people and young women. If we can return them to a normal life, and it will be transformative for them. And it will also be transformative for society and our medical systems because these people suffer for 20, 30, 40, 50 years. And I'm pretty optimistic that there are going to be a number of them that are going to be able to wave goodbye to these diseases for a prolonged period of time.
And Tom?
So I'd say three things. Three things made me realize that this is really a big, major changing point in medicine. First, when I was in Boston at Mass General, we worked a lot in lung cancer, spent a lot of time in the area of targeted therapy, and we did a lot of work with EGFR and ALK, and at the same time, BRAF was being developed in melanoma. And I was at the MGH at the time working with Daniel Haber, and the idea was it's all about signal transduction. It's all about understanding pathways. We'll find pathways that will make a big difference. These immunology guys are a little crazy. Okay, and we thought we kind of had the road to curing cancer. So I leave there and go to Yale, and I meet a young guy named Scott Gettinger and Mario Schnall, who are two clinicians at Yale, and they came up. I was the new cancer center director, and they came in and they said, we are doing immunology research. And I said, that's nice, but it's all in signal transduction. We're going to profile everybody. We're going to find cures for cancer. And they said, well, do you want to just take a look at a few CAT scans? And Scott was one of the original nivolumab investigators in lung cancer. And I was a lung cancer doctor. I had never seen the responses to the PD-1s yet. And Scott started showing me some of the CT scans. And I was absolutely floored. The depth of response that they were seeing with Nivo, a PD-1 drug, and I have nothing to do with Bristol Myers anymore, so I'm not saying that Nivo is any better than anything else. Could be Pembro, could be Atezo, but it was absolutely remarkable. So that was number one. Number two were 2 days ago. New England Journal came 2 days ago. Neoadjuvant Ipi-Nivo for melanoma, stage 3 melanoma. Study was stopped after 8 months, 2 years, stunning differences in progression-free survival by getting upfront therapy. And the third happened. I'm a tennis player, not a very good tennis player, but I like to play tennis. I enjoy it. I like the competition. After we play, we sit around and have a bunch of beers. Okay, that's kind of social. It's Seattle. It's a little more social than other places. So anyway, sitting up there, having a beer with an anesthesiologist. So my buddy Dan, and I happened to whoop him, Steve, unlike when we played golf and you whooped me.
We did things together in Seattle.
But I'm sitting there with Dan Warren, an anesthesiologist, and we're chatting. We have a beer. We have a second beer. And we're talking about our careers. He's an anesthesiologist. Said, so, tell me about anesthesia. What are your favorite cases? Tell me what you do. And he said, well, Tom, he said, I do a lot of big cases. And he said, and this was unprovoked. I didn't tell him I was coming to talk at an immunology conference. And he said, I used to do a ton of IBD surgery. He said, I really like those cases because they were in young women who had horrific Crohn's disease, and we would be taking care of fistulas and doing all kinds of stuff. And he said, I haven't done one in about 3 years. And it dawned on me, oh my gosh, these same pathways that were making such a huge difference in cancer were transforming the lives of men and women with Crohn's disease and ulcerative colitis. And so you see the other end, Priya, as you're pointing out, that is incredibly important. Now, I think one thing I hope we get to today is the balance between the two, and how many of you as investors and pharma folks, how you're thinking about those two, balance between autoimmunity and cancer. But it really is a remarkable time.
Thank you. Maybe let's just go to talk a little bit about the therapeutic modalities and some of these new modalities that have emerged that allow us to leverage the immune system. I'm going to start with you, Steve.
For me,
Yeah, for you.
modalities. So maybe I'll just start with what I tend to spend a lot of time on, which is cell therapy, right? And what is so beautiful about this modality is it actually is living and it reacts to the environment that it lives in. And it has probably the best opportunity to date of having curative intent, right? And almost every disease we know of is caused either by a missing cell or dysregulation of a cell, right? And so to the extent that we can replace a missing cell, it can be really transformative. To the extent that we can reprogram cells or replace them, the damage, once again, it can be transformative. I'll give the example of type 1 diabetes, which is a very simple autoimmune disease, very complex but really simple to describe autoimmune disease, which is your immune system attacks the beta cell, which is the cell in our body that makes insulin. And up until 101 years ago, 1923 patients just died when they had type 1 diabetes. And then there was the discovery of the ability to isolate insulin. And patients have done increasingly better, but it still is a disease where patients have about a 15-year shorter lifespan than a non-diabetic. They have to make, on average, something like 140 executive decisions a day. What am I going to eat? How am I going to exercise? How much insulin do I take? And these can be life or death decisions. Am I a little sick today? And in that time, they also might lose their kidney or have blindness. So it's complex every day. It's got lots of complications, and it leads to a shorter lifespan. And imagine if you could just replace that cell, right? So just replace it. And now you have a glucose-sensitive insulin-secreting cell, and it's a single transplant, and the patient can go and live their life as normal. So I'll give you we have done that in non-human primates. I'm hoping that we'll get that done in humans in the next few months for the first time. And if that does happen, again, that is another tipping point for medicine, right, around the ability to make cells. Turns out you make these from stem cells. The idea of making gene-modified stem cells that you can transplant and really kind of have curative intent for patients is something that is, I think, very powerful. And it speaks to why, even though there are tons of challenges with new modalities, in the 1990s we couldn't make antibodies. They were too expensive, they were too hard to give, there wasn't the infrastructure to deliver them. Today they're the largest therapeutic class in the world by far. The same things hold true today for cell therapy. They're too complicated to manufacture, they're too expensive, and we don't have the infrastructure really to deliver them. And it's all three of those things that have to be solved. But if the science really works out like we hope it will, I'm quite optimistic that the hospital system, the payer system, and our ability to manufacture them will all evolve in a way that we can again offer patients curative intent.
And Priya?
I'd love to talk about multiple sclerosis. As you know, this is affecting 2.8 million people or so just in the United States. It's very debilitating, can be relapsing, remitting, can be progressive, multiple forms in between. And I'm really proud because Biogen as a company has brought so many options to patients.
Pioneer in the field of MS.
Thank you. Thank you. We're proud of that legacy. But I think I want to double click on the modality question that you asked, Lynn, because when you look across the spectrum, and I'm going to go from 1996, when Avonex got approved, and this is an interferon beta 1A, and then, fast forward it to a pegylated form, which is Plegridy, and then in between you have Tysabri, that's a high efficacy but causes a serious brain adverse event called PML, but still on the market and doing quite well. And patients who are on Tysabri never want to leave, actually. And because of them, we brought it back to the market when it was withdrawn voluntarily. It addresses, it's a monoclonal antibody, and it addresses adhesion molecules that prevent the leukocytes from getting into the brain. And then we've got small molecules like dimethyl fumarate or Tecfidera, then Vumerity, and these are acting by a completely different mechanism of action. And Steve, when we were talking, you said something really profound the other day, that, how is it, just for us to step back and think about complexity of disease biology is very important. And we talked, you mentioned MS, and the fact that Tysabri, which is an anti-VLA4, blocks the adhesion, but Ocrevus, that is an anti-CD20, works very well as well. How do you put all of these things together? So I think we are, in some ways, I know I said that I think it's going to be a decade of breakthroughs, and I believe that. But in many ways, there are lots of opportunities. There's a lot more to discover. Because this is complex. There's a lot of crosstalk amongst these mechanisms. And what I hope we're eventually getting to very soon is a way in which we can digitalize the immune health of a patient to be able to predict what medications for their given disease they'll be able to react to best. So a prognostic indicator. And I think we're getting there, but this is the observation just across our own MS portfolio makes me believe that that is true.
Yeah. And I like MS as an example too, because it crosses through the autoimmune to the neurology, brings the two together in the neuroinflammation, and it was discovered in 1868 and still so highly researched, but we still have nothing for progressive MS. So really important, and as you point out, the different mechanisms that have been with these two different drugs, so really still not a complete understanding of the biology.
Far from it, I would say, far from it.
Yeah. And then I want to come back to immunotherapy and cancer. So as you know well, Tom, cancer is the second leading cause of death worldwide. They expect to see record-breaking number of new cases in 2024 here in the U.S., with 2 million people being diagnosed with cancer. It's estimated 600,000 of those will die. Immunotherapy, obviously, is fundamentally a very different approach to cancer treatment. It's unlocking the body's own immune system to fight cancer. You've been at the forefront of this for many years as a physician scientist, and including at the Hutch and what you do at what you did at BMS. So talk a little bit about where we are today and how IO has revolutionized cancer therapy, and has the promise lived up to what our expectations were?
I will. Can I just say one thing about MS?
For sure.
So follow me on this weave. All of you need to perfect your weaves, okay? As you're talking about MS, I thought about the impact MS has had and immediately went to John King. John King being the CNN reporter who was at the big wall during the election returns. My government and public affairs people said, do not talk politics at this panel. I am not going to talk politics, okay? I'm talking about MS. But this is the weave. Okay?
We're watching the weave. We're watching the weave.
You're watching the weave. So I'm watching John King, and I'm thinking how remarkable it is what he's able to do 25 years into a diagnosis with MS. He's been very public about his diagnosis. And what dawned on me was that in this room, I'm sure there are many people very happy with the election, many people concerned about the election, but the reason that he was able to do that is because of the investments that all of you in the equity markets have done, as well as what the government has done in immunology. And I think we're at a really critical point. And you spoke about Biogen. I'm going to just speak from the perspective of running a science research facility. The NIH funding that powered so many of the discoveries that you're going to hear about throughout today's conference comes from fundamental basic science research. Last year, the NIH budget under a Democrat went down by 0.8%, not inflation adjusted, absolute decrease in NIH funding. Next year, we'll have a Republican administration. It is the duty of everybody in this room to advocate to keep the NIH budget at a place where we can still innovate. None of you will have companies to invest in, or companies to found, or companies to run if we do not invest in the fundamental basic science. This is not a Republican problem. This is not a Democratic problem. This is an American problem that we've got to solve. We've got to make these investments. And when you see John King and see the remarkable impact on his life, and I'm not sure which Biogen drug he's on, but I suspect it's several. He's been on several, I'm sure, along the way. When you look at what has happened to him, and you realize that came because our government invested in fundamental science, which allowed the development of many of these new technologies. So I just wanted to mention that.
Yeah, no, it's great. And IO, I'm sure the same, right? I mean,
oh, and then same thing in cancer. I mean, it's on both sides of the same, curve. I think from a cancer standpoint, what we're most excited about at the Fred Hutch, I'd say, in immunology are a couple things. One, we've got a large viral immunology program that's working on COVID and HIV. We have a big HIV vaccine group. There is still a need for HIV vaccines. I know that may sound crazy with some of the great long-acting anti-HIV medications we have, but when you look at where HIV is a problem, it's a global problem in sub-Saharan Africa and other areas where even taking a medicine every 6 months is not something that's going to be feasible in those areas, and a vaccine offers tremendous benefits. So that's one area of immunology that we're investing in. On the cancer side, it's really about trying to figure out how you can take these incredible responses that we're seeing in myeloma and B-cell malignancies and melanoma and start to see them in solid tumors. Now, we've seen some of that now in small cell lung cancer. We're seeing antibody-drug conjugates and T-cell engagers making a difference in those areas, but it's really about trying to get immunologic activity to work outside of the classic immune-related diseases.
That was Muhammad Ali-like weave.
There was a weave there, yeah.
I know, I was a little scared for a while. All right, so let's talk about the market opportunity and the challenges. So the I&I space is predicted to become the second largest area of biopharma spend, behind oncology by 2028. The expected revenue this year for approved immune-based medicines is around $100 billion. That's expected to double over the next 8 years. The M&A transactions of immunology-based companies and platforms of at least $1 billion or greater have totaled $71 billion in the last 4 years, starting with 2020. Biogen just made an acquisition in 2024, in May of this year, of HI-Bio. So why did you do that? Why did you acquire them? And then talk about the scientific platform as well.
Sure. And I'll just step back to say that, I think oncology and immunology have a lot of similarities because they manifest in different system organ classes. And what could work for the mechanism in one system organ class could have some extrapolation to others. And that helps us learn, right? So I think that's really critical. Now, when I first met Travis, the CEO of HI-Bio, he talked about HI-Bio as an autoantibody company. And that's what intrigued me. Because, even with the fact that I'm absolutely not an immunologist, I have a working knowledge, specifically as I think about therapeutics, about the relevance of autoantibodies in disease. And then he talked about, exciting phase 2 data. And, of course, we did a lot of diligence. Our teams did a fantastic job diving deep, and HI-Bio really partnered with us, shared all that data. But what was really exciting was the elegance of the mechanism of action. So here's felzartamab, that is an anti-CD38. And CD38 is expressed on plasmablasts and plasma cells. And when you tackle those, you are able to address bringing down those antibodies, but not impacting the immune system's ability to really defend against virus, disease, all of that. And they actually showed that with data where they showed in IgA nephropathy, which is one of the 3 indications, and I'll talk more about the indications, but this is really cool, because they showed data that with 5-month therapy, and it's a specific regimen, at 6 months, you see everything kind of drop, IgA, IgM, IgG. But slowly, the IgM and IgG rebound back, whereas IgA remains down out to 24 months. That was intriguing, because I think we're all looking for this sort of threading the needle, the right weave, so to say, to really address the bad actors, but not prevent the good actors from coming to the forefront and defending the body. And so that was very intriguing. The other piece here is that, as we already talked about with multiple sclerosis, all of these other areas that we've been in, lupus, including lupus, where we've got 2 phase 3 programs, what's exciting to us is the immunological basis for disease. And we saw the promise in high bio with the anti-CD38 sort of mechanism of action. And importantly, as we were in the real important evolution of ensuring that we were thinking about growth, that we were being very disciplined about the aggregate risk reward of our portfolio, that we were investing in programs where we had a reasonable degree of confidence in scientific conviction in phase 3 to really bring value. And that's what clinched the deal for me personally and for my team that kind of did the diligence here, because we saw proof of concept not only by biomarkers that were predictive, but also in clinical data. And we saw these 3 as phase 3 ready. So we felt that as sort of the drug development chops that we bring to the table, this would be a wonderful combination of the science, the nimbleness in which High Bio thought about their platform, applied it to 3 nephrology indications, but then really thinking about how do we take it to the next level and help them with our unique expertise. So that's what I think it kind of, it worked on all sides, and we're really excited. We're starting 3 phase 3 programs in 2025, all high unmet need, antibody-mediated rejection, IgA nephropathy, primary membranous nephropathy, the commonest cause of nephrotic syndrome. And we're looking at a lot of other indications where this mechanism might be relevant beyond the kidney.
Yeah, amazing. The opportunities are so great right now for I&I. One of the stats when we were preparing for this, 189 I&I programs in phase 2 and 3 across 8 therapeutic areas. That's huge. 60% in development for one indication, 40% for multiple indications. So as we think about the opportunities, obviously there's a lot of challenges there, and some of those you mentioned, Steve, but what are some of the other challenges that we really have to overcome if we're to go to that curative intent in the next decade?
Well, yeah, that's a good question. A couple of things I was thinking about. Well, if you take big platitudes of time, you had a whole period in the pharmaceutical industry called the 80s and 90s where value was accreted to programs and companies based upon the size of the market they were going after, right? And we then went through, I think, a period where the value really accreted based upon the size of the benefit that was with the therapeutic. But it ended up that a lot of those therapeutics went after then smaller diseases and orphan indications. And now I think we're hopefully in an era where those 2 things come together, and where the size of the market really matters, and the size of the benefit is essential as well. And what you have, though, is you have very crowded marketplaces, right? And one of the things we have to really overcome as an industry is figuring out, this coming out of big eras of increased cost of capital and pressure on our system, or I should say on our ecosystem. If you go back again, 2002 to 2008 was an era of specialty pharma where it was a bunch of Me Too drugs. And one of the concerns I have looking at the industry today is it's a bunch of Me Too drugs, right? I mean, you have multiple companies with the same modality or different modalities going after the same target, right? And so you have a world that's still very, very risk averse, right? And to really make a big impact, we're going to have to take risk and go into new modalities, new targets, and new biology. Because every single clinical trial you try to run right now you have a couple of challenges. And one of them is that there's huge competition for space within the places that run clinical studies. And in fact, most of them have faced what we used to call the Great Resignation, right? They haven't yet recovered from that, and their clinical trial infrastructure remains, I'd say, at best, back to baseline. The second is that when you're moving into new modalities or different side effects, you end up taking up more infrastructure space, right? I think that's been a real challenge for again, bringing these medicines into patients in clinical studies. And the third is there's huge competition for these patients, right? And so I know a lot of people here are probably very excited about the impact of artificial intelligence. I love that, you could have a Nobel Prize go to both, a great scientist and a great person like David Baker. But where we really need to kind of make the investment in artificial intelligence is not in making binders. We're pretty good at that, right? We're pretty good at making that stuff. We really need to understand both where better back to the basic biology and find novel targets. Because of those, I would bet you if you took that down, you'd say there are only like 183 drugs in phase 2, probably 18 classes or 15 classes or 10, right? And then we need help in clinical development and getting the right drug to the right patient at the right time. And the big bottlenecks in the industry are not making the binder. It's finding the novel biology, and it's the cost and time of drug development. And we really need help in those two areas more than anywhere else. I don't know. That's a weave, I guess. But.
Yeah, so I guess one question I would ask both Steve and Priya is that one of the things that worries me is the relative investment in cancer versus inflammation. And it's a shame to think of them as having to be a trade-off between the two. What I worry a little bit about is that pharma and biotech's appetite for cancer has gone down recently, and we're seeing less investment in cancer. The trials take forever. The endpoints are challenging. The science is difficult. Creating CAR-Ts that work in solid tumors is really challenging. T-cell receptors based therapies are really narrow, tiny little markets. And yet what I fear is that taking the foot off the gas at this point, when we're so close to breakthroughs in so many cancers that we haven't seen, I worry that people are saying, gosh, what about a seventh therapy for psoriasis or an eighth therapy psoriasis? Because it's such a great market, and those patients will have psoriasis forever, and there's so many of them. It's really, I worry about the competition for investment dollars between cancer and autoimmunity. And I guess the question is, for the two of you who run such important companies, how do you think about that? And should I be worried as a cancer center director, or do you think that this will work out?
I'll start with an answer that you're not going to like. Earlier this week, we announced that we were shelving a cancer drug and putting more of our resources into different inflammation targets. And so, and it had everything to do with the competitive nature, prolonged timelines, and necessity to run big randomized trials, which is very difficult for a relatively small biotech. And so I think you're justified in being worried. And I do think we're on the precipice of some really important progress in the oncology space, and it's become more and more complicated to run these studies, to fund these studies, and that's not a great thing for patients.
Do you think the trials are more difficult in cancer than some of these chronic diseases?
Say it again?
Are the trials, are they more difficult in cancer than some of these chronic autoimmune diseases? I mean, it always seems to me the diseases, the chronic diseases, you have to go all the way through the phase 3. So it seems like that would be more time, more money. But what I'm hearing you say is some of these cancer trials are more difficult.
Well, there are complexities in both. Let's not kid ourselves, right?
I mean, it's not either or, I realize. Yeah. I was just curious.
Yeah. Just a couple of points. It's a great question. I think I come at it from a slightly different perspective. So I think overall, as I mentioned, one of the statistics that worries me is that while autoimmune diseases are 3 times as common, there's this drug development, at least, in that space is 7 times lower than oncology is today. Having said that, I agree. We can't take our foot off the pedal. as an industry. Now, when I zero in to what we do at Biogen, I must always have the lens of what are we good at? Does the world need us to do it? And can we make a dent in it? And those are the three questions that I and my team and our organization, we keep asking ourselves. Because we're looking at how do we bring value to unmet need, patients with unmet need. And I want to bring in this factor that you brought in about the chronic trials. So I think oncology has some real advantages, which I hope we don't lose in terms of regulatory flexibility, accelerated approval, expedited pathways. We, at Biogen, we really did not see that benefit translate over to a disease area like Alzheimer's disease at all. But I think that the burden of disease, you could argue, is very similar. So I would say, and when it comes to clinical trials, I will tell you we were recently reviewing our phase 3 trial for litifilimab, another immunological pathway sort of interruption with the type 1 interferon signature through BDCA2. And we were looking at this and we looked at the list of lupus phase 3 trials that are ongoing. There's more than 30, 40 in phase 3. So we're competing with all of them. And it's very hard to recruit. So I think very hard, and the regulatory flexibility is very limited. We have to have 2 trials that replicate, reproduce, all of that, and they have to be fairly large.
Do you think you have an infrastructure for that? So in cancer, you've got cancer centers, you've got
Yes.
an international group of people, investigators working together. Is the infrastructure as good in inflammatory disease?
Absolutely not, is my view. I think that the cancer infrastructure is something that, and the way oncologists approach research at centers around the world, I think is vastly different from how they treat any other disease. We faced it in clinical research as well as translation of molecules that get approved in the commercial setting. So I think it's a real problem. And I think that's where we need to really think about what are the lessons that we can transfer and generalize, because I think the problems are at every level. Oncology for me is kind of a blueprint, a role model. How can we do that for patients with unmet disease, unmet need across so many other disease areas? It continues to be a conundrum.
Hey, Tom, I had one more thought on your question, which is, again, without getting into politics, government intervention tends to have unintended consequences, right? And so with the Inflation Reduction Act, you have a new way of a new cap on how long drugs in the United States will have basically pricing power, right? And so there's a roadmap for developing drugs in oncology. And that roadmap is you go into end-stage cancer patients where there's a small but defined unmet need, and you move and invest to move that therapy up further and further into the treatment paradigm. And the value for the patient, the value for the company becomes greater and greater as you move into earlier stage therapies. But if you only have 9 years from the time of approval, that's broken. It's absolutely broken. And that doesn't mean that we won't fix it, that there isn't a different way to develop these drugs. But whenever you kind of break a system, I think the industry is going to have to figure out how are they going to develop drugs with a reasonable ROI within the context of a totally different reimbursement environment. And that will be a shock, and it's a shock we're going through right now that we'll have to work our way through. But you can't do it the old way because,
I was reading a really interesting statistic based on the IRA that while there's an orphan drug exemption, it's only for the first indication.
Yes.
And a study has shown, quite robustly, that the return on investment for any orphan drug on its first indication is only 19%. And everything else comes from the follow-on indications, which are no longer incentivized. So it's another.
Well, and you can't do it in the sense like you did before, where you have the first and then you bring up,
Exactly. So lots of patients.
to almost put them all out there at first, right? Or the clock is ticking and you don't get
Exactly.
the value.
Yeah.
It's a real issue. I guess just coming back for a moment to the regulatory part of this, is there anything, that you would hope that the FDA, you mentioned in oncology accelerated approval and those types of things that you hope the FDA would consider or should be considering in the area of I&I with those types of trials?
I guess one thing that shocked me when I was at Bristol Myers and suddenly was dealing with things outside of cancer was just how much more attention was paid to toxicity in chronic inflammatory diseases compared to cancer, as it should be, okay? But the attitude, you talk to a cancer drug developer and you get 3-time elevations of liver function tests, it's like, okay, no big deal. Somebody's got psoriasis and you're going to bump their LFTs up 3 times above normal. You can't do that, okay? And the drugs don't get developed that way. So there is a big difference in how one looks at toxicity appropriately. But that same ability to be relatively toxicity averse, less toxicity averse in cancer has helped develop drugs quicker.
Yeah, go ahead. You go, Priya.
Please. From my perspective, I think there is a potential key to unlock this problem. And I think it will come from all the work, the basic science, the ecosystem coming together. And I think it's about biomarkers. Because I think that the FDA, I've been always very impressed in reviews about the questions they ask us, how thoughtful they are. They're really trying to balance the benefit risk. And I think that the beauty of oncology drug development has been in the ability to molecularly type the cancers, classifying tumor neoantigens, all of that. That sophisticated work, I think, is still early in the chronic inflammatory disease space. It's picking up, and I think it's getting there, but it's not there. And I think FDA and other regulators around the world, they need something that is reasonably predictive to hang their hats on. Because otherwise, we're also going to have a lot of drugs that will not benefit patients. And we don't want patients to be the sufferers at the end of the day. We want them to be the winners. So the question is, how do we balance this? And again, I think there's a lot we can learn. But if we can come up with some of these biomarkers, and I'll give you just one example. In progressive MS, that is a black box for us. So today, investing in progressive MS means you're going to have to take it out to 10 years. You're going to have to spend $1 billion and get to a phase 3. And there are no bets on whether you would succeed or fail. But if you had a biomarker, you would be willing to invest till you can get to a de-risking stage. And we're not there. That's what we, I think, need, which we can never get to without the ecosystem of, basic research and all the parts kind of coming together.
Steve?
If I had a message for regulators or wish, it would be to listen to patients. And I think that there is a tendency that I've seen in these chronic diseases to underestimate the toxicity, all that happens to patients over 10, 20, 30 years. And that relates both to the disease and to the therapies, right? And so if, for example, a bispecific antibody that Priya mentioned could take a patient and get them into a drug-free remission, there's a trade-off. There might be higher short-term toxicity, but they're getting rid of decades of long-term toxicity, both from the disease and from those drugs. And I don't think that we are very good at evaluating that. We look at them as if the 1 year of your TNF inhibitor is equivalent to that very brief period that a patient is on a therapy. And I think we're way underestimating the potential for curative type therapies and to get patients off of drugs and give them a long period of normal life. And that's what they want desperately if you listen to them. And I think that's true in almost every disease we talk about. And so maybe force us, who are trying to do curative therapies, to really define our side effect profile and then give it to the patient to decide. Let them decide. This isn't for the government to decide. This isn't for me to decide, let the patient decide.
If I can throw one question, Steve, just listening to you talk about the concept of toxicity. One part of toxicity from another part of my life, which is running a cancer hospital, is the management of toxicity and the cost burden that's now placing on the healthcare system with immunotherapy. So the Hutch has been someplace we pioneered outpatient allogeneic transplant. We do a lot of our allogeneic transplants as outpatients. We do our leukemia induction therapies as outpatients. But with the proliferation of CAR T-based therapy, TCR-based therapy, even the bispecifics and the T-cell engagers, those people often need hospitalization. And it's putting pressure on our hospital bed capacity. In the state of Washington, we have half the number of hospital beds per person as the state of Massachusetts, similarly sized states, and we don't have the hospital capacity. And we're at a time now where building hospitals is unbelievably expensive. The cost to build, I mean, as we all know, we have an enormous housing crisis in this country because of the cost of building that's become so expensive. Building hospitals, particularly on the coast, is really, really challenging. And so as we come up with these therapies that do require, I mean, you give somebody a CAR T therapy or a TCR, many of them who get cytokine release or neurotoxicity don't know their name for 2 weeks after treatment. Not all, but many. And you've got to have the facilities to support them in that setting. So there is that unintended cost that people don't necessarily bake into the cost of delivering the drug, but it is a cost to the healthcare systems as we go forward.
Tom's building a hospital, so if you guys want to donate, he's all up for it.
Always looking for people interested in supporting cancer.
There you go. Okay, so maybe just to come full circle, do you think in the next decade we can find the cure, we can reset the immune system? Can we get there, our curative intent? Steve,
Well,
you mentioned
a cure,
George Shedd.
absolutely not, right? A cure for some people, I think it's almost certain, right? And so, the good and the bad, I think, is that these medicines are already in development. They work. Patients will have long-term drug-free remissions. The bad is it will not be anywhere near 100%, and we're going to have to learn. And this is why there are some Me Too's and multiple modalities going to have the same disease. These are complicated diseases. And why is it that, as Priya said, a B-cell depleter and a T-cell depleter both work in multiple sclerosis? Is that the same drug? Why is it that in rheumatoid arthritis you have these TNF inhibitors that are so potent, but then you also have these B-cell depleters that seem to work so well? They're like completely different mechanisms, and they have, so these, I think these are mostly syndromes, right? And we call them diseases, and we're going to have to get better at defining the diseases so that we can get closer to, and by syndrome I mean something that has a clinical manifestation, and a disease is a single pathology, like a pathway. And so we're going to have to get better, and we will have cures for some diseases. We will not cure all of these syndromes.
I don't know, Priya, Tom?
I think I agree completely with what Steve said. I think we're going to be able to do it for small subsets of patients, but we may not be able to do this for all. But I think there's another promise that the decade could potentially unlock. And that is that for some important areas, we may be able to immunotype and really focus on the immune health of the patient and figure out what therapy is going to be better for them. And I think this was one of the few boons of the pandemic, because today, if you have an elderly person who has COVID-19, you could immune type them, and you can see whether they're low on T cells and they need a vaccine boost, or they really actually don't need anything and can go out freely, or they need passive antibodies because they're not making enough antibodies. And I think that is pretty revolutionary. If we could apply that to other diseases, I think we could do a lot more in terms of predicting, and that would help us in the clinical trial setting as well, because we'll be able to patient select better. That means we can run better trials because we're not going to be looking all for the same types of patients. And so I think it has a lot of positive consequences.
Can I add one thing? Because I kind of beat up the people who were doing the same thing over and over again, but basically the B-cell depletion field was started in the mid-1990s with Rituxan, right? And we're still making, there's a huge industry that's based upon better B-cell depleters, right? And this is what will drive curative intent, right? You get Control-Alt-Delete of the patient's B-cell repertoire. It's no different than what you do with a computer, and it comes back and it fights disease, it fights viral infection, but the pathologic antibodies are gone. And so, there is something to be said for, continuing to invest in, known mechanisms until you get the definitive therapy. And clearly, there's been tons of progress and better patient outcomes because of people who continue to invest in great B-cell depleters. Go ahead, Tom.
Yeah, I just say, I'm a medical oncologist. I still practice. I practiced intensively for 25 years in Boston and New Haven. And I'd say a couple things. Oncologists are optimists. We spent a lot of time talking about problems today. Low funding from the NIH, challenging side effects, challenges within companies about whether to invest in cancer versus inflammation. Let's just stop and remind ourselves, folks, the system is working. We are making progress. The ecosystem that supports progress in cancer and in autoimmune disease is working. I keep coming back, and this is something I mentioned with Steve when Lynne had us on a panel in Seattle. I think back to my first experience in cancer. My first experience in cancer was working as a lab tech in my father's in our basement of our house, where my dad had a hematology practice, and I would I would spin the hematocrits, and I would prepare I did it without gloves too. I would make the right smears, and I would do a white count, and I would do a differential, and I was 12 years old. But the key thing is, I would watch the patients come in and out of the office. And at that point, I would say at school, my dad's a doctor, he's a really good one, but most of his patients die. Because that was my impression, okay? And if you think of a patient diagnosed with myeloma in 1972, when I was making those smears for him and seeing the rouleau of the red cells, 12 years old, doing pretty well. But the point being is that those patients lived 18 months with melphalan and prednisone. And then when I started my career in 1992, we were still using melphalan and prednisone, and they were living 18 months. If you're diagnosed now with myeloma, you can expect to live 10 to 18 years with all these therapies. So while we might struggle with the problems, and of course, if we don't recognize the problems, we're not going to improve things, the progress we've made in basic science, the progress we've made in cancer centers at biotech and at big pharma, fueled by much of the investment that many of you have helped enable, it is making a difference, and we will continue to see incremental cures in cancer and in inflammation.
I give him a hard time because as a lung doctor, all he had to do in training was learn Taxol versus Taxotere, right? And now you've got to actually really dig in and understand a lot about this disease, and there are a lot of different modalities, and a lot of patients do a lot better.
I'm going to come back to my first point, which is we are at the tipping point. It's going to be a revolution.
Thank you so much. You give us hope. We believe deeply at Alexandria in medical innovation in the life science industry, and we thank you for your commitment to medical innovation and advancing human health. So let's thank our panelists, and thank you so much.